Okay, let's start. Thanks so much for the invitation. I'm going to talk about the motivation, about the data. I don't know if it's familiar to everyone, these kind of Hi-C and HiChIP data sets I'm going to talk about, and also talk about predictive models of gene regulation, which has been an interest of our group for a long time. So I hope everybody has this idea in mind, this picture of transcriptional gene regulation. So, as we know, there's an important kind of proximal regulatory information at the promoter close to the transcription start site, where you have binding of transcription factors that enhance initiation of transcription. But there are also, maybe I should I do a laser pointer? Okay, I don't know if that's clear. There are also these more distal elements. They can be in introns, they can be in intergenic regions where you also have binding of transcription factors. And these regions, while far away in the 1D genome, maybe more than a megabase away from the promoter, they loop and interact with the promoter and enhance transcription. Okay, so these more distal enhancer elements are critical for the regulation of many genes. And for a long time, we've had 1D epigenomic data like chromatin accessibility and histone marks that allow us to map these regulatory elements, but we didn't have good information of their connectivity, and that is changing now. Okay, so just to motivate this work, we and many other people have spent a lot of time looking at differentiation programs in different contexts. So I'm taking this figure from my own work, but there are many such studies by many groups. Okay, and in this case, we're trying to look at, understand how CD8 T cells differentiate to dysfunction in the context of tumors or chronic viral infection. Okay, and what you can see is I'm showing you the locus that encodes PD1 and another locus encoding interferon gamma in the mouse. Okay, and what happens as you differentiate to dysfunction is the accessibility landscape here mapped by a TAC-seq changes. And somehow these changes are encoding that you gain expression of PD1 and lose expression of interferon gamma. Okay, and in particular, there are specific events, right? So here's a famous enhancer that you only see on T cells that are on their way to dysfunction and not in normal effector memory cells. There are other sites that are supposed to be highly accessible and they lose their accessibility. Okay, so somehow this set of accessibility and also presumably chromatin state as defined by histone marks is changing how this gene is regulated. Okay, so how do we go from this map to gene expression? And we have spend a lot of time trying to combine this 1D epigenomic data and DNA sequence data to predict gene expression, okay? And the missing piece always was this connectivity of enhancers to promoters. And the idea that I'm gonna talk about for most of today is using 3D interaction data from chromosome conformation capture assays to define a graph and using the formalism of graph neural networks in these predictive models of gene regulation. And I want to make the point here that the reason we're doing this is not to accurately predict gene expression per se. Okay, so often of all these things, all the ingredients in the model, the easiest thing to measure is the gene expression, right? We're doing it because we want to understand how genes are regulated, where are the enhancers, what are the regulatory elements, what are the transcription factors binding these elements. That's the motivation. So it's a sort of a more mechanistic motivation, but we're using machine learning. Also, another important motivation is trying to understand the role of non-coding genetic variation in disease, okay? And we'll talk about this ABC score a little bit later. So this is a figure not from us, but from a recent big study from Jesse Engreitz. And this ABC score is a score that combines Hi-C data, which we'll talk about in a minute, together with some epigenomic data, like histone mark data and accessibility, to try to figure out which enhancers might regulate which promoters. And then when you have genetic variants that through GWAS were associated with disease, perhaps you can say, well, this non-coding genetic variant is sitting in in an enhancer and it regulates this gene. So you can start to do this, take associations and functionalize them. Okay, so ultimately, if you have predictive models of gene regulation, you can infer the role of genomic elements and how genetic variants affect gene expression. Okay, so I have to step back and talk a bit about mapping the 3D genome, talking about the assays and how to analyze. And the analysis is non-trivial, okay? So Hi-C is a sort of an all-by-all chromosome conformation capture assay. Basically, what you do is you cross-link DNA in situ in the nucleus. You do a restriction enzyme digest. Okay, so you have these ends, you fill the ends, mark with biotin ligate, and you pull down these glued together regions so that when you do a paired end sequencing library, you have these reads that may be distant in the 1D genome, but they were mapping regions that were close in 3D, okay? And that's a contact. So, and if you have many reads with the same anchors in your library, that's suggesting that that's real, okay? It's a real 3D contact and not just background noise, okay? So what we get from these libraries is usually visualized as a contact matrix where Cij is the number of paired end reads with anchors in bin I and bin J, okay? So what do you do with this contact matrix? Depending on the depth of the data, you can analyze at different resolutions. We're interested in gene regulation, so we're interested in fairly zoomed in part of this figure. These structures kind of called topologically associating domains. Okay, so these triangles that you can see in the normalized contact map, which we think might be self-contacting regions where genes and their enhancers are kind of organized in a domain. And sometimes with very high resolution data, you can even see these loops that might represent promoter enhancer interactions. So these can be quite subtle to see in a kind of regular quality Hi-C. Okay, so I just want to talk a little bit about this method that's now published. There's a new version called HiC-DC Plus, where we want to take the contact matrix and assess whether an interaction bin, okay, the CIJ count, is significant if it represents a real interaction or if it comes from a background from the null distribution. Okay, and we call it Hi-C direct caller because we're making calls directly from count data. We're not doing some kind of smoothing or normalization prior to the statistical analysis. So what we do is we estimate a background model directly from the data using negative binomial regression. The covariates are the genomic distance. Okay, so what's going to happen just from random polymer ligation is that the number of reads that you get decays off the diagonal. Okay, so you have to model that. That's the main feature of the data, this sort of dependence on genomic distance. But there are also other covariates like the effective size of your bin based on how many restriction enzyme sites you have, mappability, GC content. Okay, so we model these covariates and do a negative binomial regression, and we have efficient code, and it actually works nicely for HiChIP that I'm going to talk about, and we can also do differential interactions. Okay, just since this is a technical audience, this is just a generalized linear model. We're doing negative binomial regression. We have a spline basis for fitting the dependence on genomic distance, and then we explicitly measure the model these other covariates. You remove outliers, refit the model for more sensitivity. You can have a variable dispersion if you want. And then there's also just a trick to, if you have Hi-C and one condition and another condition, and you have replicates, you can correctly do differential interactions just by using distance-dependent scaling factors. Okay, so if you're familiar with the statistical analysis of read count data, that made sense, otherwise maybe not, but just to say that I think this is the right way to analyze the data and to call significant interactions, okay, rather than doing some kind of pre-processing on the count matrix and trying to find bright spots, like treating it like an image analysis problem. I think this is better. Okay. So just to show you a few examples before we get to the regulatory model part. You know, this is data from a collaborator, Danwei Huangfu. She and Effie Apostolou and I were in a 40 nucleome project where we're looking at enhanced rewiring in directed differentiation of human ES cells to pancreatic differentiation towards insulin secreting beta cells. And this is some initial Hi-C data, ES cells to definitive endoderm and pancreatic progenitors. And what you can see after you do the analysis with HiC-DC is you can start assigning significance to interactions that are getting set up around this gene PDX1. It's an important developmental transcription factor and a diabetes gene. Okay, so just showing that even though this is not super high resolution Hi-C with the right statistical analysis, you can actually see these interactions that look like enhancer promoter interactions. Okay, one more background slide. All right, so when you do Hi-C, because it's this all by all interactions, you have to do many libraries and you have to have a lot of sequencing. So typically, if you want 5 KB resolution, you need 2 billion reads. And that's expensive. So what people have done is developed approaches to enrich for interactions of interest. So one is HiChIP, which is essentially you do your Hi-C library and then do a chromatin IP for a protein or histone mark of interest. And of interest to us is H3K27 acetyl HiChIP. So this will enrich for regulatory interactions like enhancer-promoter, promoter-promoter, enhancer-enhancer interactions. Some people want to enrich for structural loops. You can do capture Hi-C. You can kind of have a library of RNA probes that you, for regions of interest. Promoter capture is of interest, or, baits along a specific locus of interest. And I think, I'm happy to report we've figured out how to do the statistical analysis of all these different data types, including recently, I think we know how to do promoter capture Hi-C. So once you can analyze, and it's, again, not trivial, you're in business, and you can reduce the complexity of the library you're trying to represent. And, so for HiChIP, you can do 200 million reads and get 5 KB resolution and enrich for these regulatory interactions that are of interest. Okay, so just to give you a picture of that, this is data from our collaborator, Effie Apostolou. It was already published. She was interested in enhancer hubs that you see in pluripotent, so in this case, mouse embryonic stem cells that disappear in more differentiated cell types. And what we're doing here is looking at H3K27 acetyl HiChIP data. Okay, so it looks a little different than Hi-C. And we're doing a differential HiC-DC analysis. And I'm showing the loops that are present in the pluripotent stem cells that are lost in mouse embryonic fibroblasts. And you see this nice hub structure, which is what she described in her paper. And it's connecting genes. So you have a bunch of genes, promoters and some enhancers all in a hub together, and other genes are left out of the hub. Okay, and this seems to be the right structure that she experimentally validated. Okay, so we've gone through the data types and the statistical analysis, and now I want to talk about graph neural networks for training predictive models of gene regulation. Okay, so the idea here is to use the 3D Hi-C, HiChIP data to encode long-range chromatin interactions as a graph. And what this does is that in the deep learning model, it gives you access to very distal elements. Okay, so you think of the linear genome just in bins. Okay, and then we connect our bins by significant edges from, for example, H3K27 acetyl HiChIP. Okay, so these are the significant regulatory interactions based on our statistical analysis. And you have promoters, you have enhancers, you have these sometimes very long range interactions, and we actually include interactions up to 2 megabases away. Okay, and now, so the nodes of the graph are the genomic bins, the edges are the interactions. Our input features are either going to be epigenomic data, histone marks and accessibility, or DNA sequence, okay? And the output is going to be the expression at each genomic bin. Okay, so you can think that if there's a promoter in the genomic bin, it's the output from that promoter. Okay, and we're going to do it over a long range, large genomic regions, as we'll see. Okay, and as our method comparison, we're comparing to kind of the state-of-the-art models using dilated convolutional neural networks. Okay, which only use 1D data and don't use 3D data. Okay. Okay, so a little bit of background. So I think this is a machine learning audience, and you're all familiar with this, but just to review, so the first class of graph neural networks were graph convolutional networks, where essentially you use the graph to encode the neighbor structure. And as you're going through layers, through the hidden layers in the model, you have your feature values at your neighbors. So for a node V, you have the previous features at the k-1 layer of all the neighbors, and you aggregate somehow, combine with the node, your node V's previous features, and that gives you the kth layer. Okay, so and maybe the way to combine is with the multi-layer perceptron, something like this. Okay, so it's just a kind of convolutional structure where you're using the graph to define neighbors that you combine with. Okay, we found this doesn't work at all. All right, but there's a more recent approach called graph attention networks that did work for us. Okay, so here there's an attention mechanism. Okay, so you have your features at your nodes. You just map into this. So you map your features into the space F prime, and you have an attention mechanism, which can just be these linear weights over, the WHJ, WHI. Okay, so this you combine, you put through some kind of nonlinearity, and that gives you an edge weight, which allows you to, again, to combine the features from your neighbors. And often, so you're kind of learning this attention mechanism, which allows you to learn how to weight the edges. And often you have a multi-headed attention mechanism. So you have multiple ones of these sort of attention mechanisms, and you concatenate them together. Okay, so the important thing here is that you're learning to weight based on the features of the at the nodes. Okay. Okay, so and we there's some we're not using exactly what's in the slide, what was in this original paper. There are some modifications that we can get into if you want. Okay, so let me explain the model, the paper. There's a version of the paper in bioRxiv that you can download. There are actually two versions of the model. Okay, so the first model is, so we call this GraphReg. I don't think it's in all my slides. GraphReg, and there's an epigenomic version and a sequence version. So the epigenomic version, we're just taking a kind of minimal set of 1D epigenomic data, so chromatin accessibility like DNase or TAC, and a few histone marks. We pass this through a few convolutional layers to learn local features from the histone, from the chromatin marks. And then we pass through the graph attention network layers that allow us to pass information from distal enhancers to promoters. And the output we're trying to predict is HiC, which is a tag-based a gene expression that also maps transcription start sites. So you can say this model predicts gene expression from the activity and connectivity of regulatory elements. It's also cell type agnostic, okay? So meaning that you could train in one cell type, and then as long as you have the ingredients, the inputs, so that means cell type specific 1D and 3D inputs in the new cell type, you could, in principle, use the trained model in the new cell type. Okay, but you would need a new graph and new 1D inputs. Okay, and that actually works. Okay, so just to give a bit of a feel for this, we are training on 6 megabase regions. okay? And binning at 100 base pairs, and we're using just 3 epigenomic inputs, DNAs. HCK4 trimethyl is a promoter mark. It kind of tells the models what bins are like, are likely to be promoters, and acetylation, okay, which is an enhancer mark. So we pass through these, CNN layers to learn local representations, and this is the graph attention, which is at a 5 kb binning, because that's the resolution of our HiChIP data. And then we're outputting gene expression, cage-seq predictions, again at 5 kb resolution. We're using Poisson loss on the middle 2 megabase region. So, we're inputting 6 megabases. We have graph edges that are up to 2 megabases away, and then the middle part of this region can see all the interactions from the whole 6 megabases. Okay, and this is just showing that if you train the model and assess on held out chromosomes, you can predict gene expression pretty well. So this is in mouse CS cells. This is predicting fold change. So where you're training the model on 2 different cell types and then looking at the real fold change versus predicted fold change. Okay, so this works nicely. The other model is a sequence-based gene regulatory model. So here there's kind of 2 tasks or 2 paths through the model. The bottom part is essentially a dilated convolutional neural network. So you have your input sequence, you have some CNN layers to learn now local sequence features, motifs, and then a dilated convolutional neural network to increase the receptive field. And you're trying to predict now the 1D epigenomic data. And the other path through the model takes these motif representations, puts it through the graph attention network layers. So now we're passing information based on sequence, and we're again trying to predict gene expression. Okay, so we are, so the bottom part of the model is trying to use the epigenomic data to guide the CNN to kind of learn meaningful representations for this cell type that will help us predict gene expression. So you can say this model is predicting expression and the 1D epigenomic signals from genomic DNA and 3D connectivity. Okay, this is definitely a cell type specific model. We wouldn't expect it to generalize out of the cell type it was trained in, because it's capturing transcription factor binding signals specific to the input. Okay, and I don't have to go through details of the model. I just, seeing that David is on the call somehow right for the slide. The bottom part of the model is quite similar to the Basenji model that from the Kelly lab. Okay, so the sort of CNN and dilated CNN layers to go from sequence to predict these histone marks and accessibility. And it's the top part of the model that's novel, that uses the graph attention layers to predict cage. Okay, so yeah. And I'll just say this is harder. Okay, so if you do the same kind of assessment, training in a cell line, holding out chromosomes, you can see that the performance is not as good, but it's still reasonable. Okay, and again, predicting fold change if you train on 2 different cell types and try to measure the expression change versus predicted expression change, at least there's some a reasonable correlation. The other thing I'll say is sometimes we segregate the results into all genes or genes that have, for example, 5 or more loops, interactions, and often the performance is a bit better relative to dilated CNNs on these kind of highly regulated genes. Okay. And again, so this is a bit of method comparison, comparing to a 1D dilated CNNs with the same inputs. So either the epigenomic inputs or the sequence-based models. So, you know, the main difference between these two approaches is use of the 3D interaction data or not using 3D interaction data. And we do see a nice boost. So if you look at expressed genes with at least 1 chip HiChIP edge or 5 HiChIP edges, you get a nice kind of performance boost. This is correlation, Spearman correlation, both for the sequence and epigenomic-based methods, and also for this kind of prediction of log-fold change, a sort of significant improvement. Okay, so this is good. We see some performance improvement over a baseline 1D dilated CNN. The sequence-based prediction is more difficult, but the prediction of expression per se is not the point. Okay, so we really, you know, expression is easy to measure in many contexts. What we want to do is interpret the model. Okay, so when you look at these models, right, so I'm showing K562 data, I'm showing the inputs here, okay, so different histone marks and accessibility. And I'm showing the HiChIP interactions where you can see this kind of looping structure and certain hubs of interactions. And what we can do is do feature attribution using saliency or, for example, deep SHAP to try to understand for each of the input features, where is the information that helps us make the prediction? Okay, so for DHPS, so this gene, we can do, for example, deep SHAP for each of the input tracks, and then sum the attributions, and we get kind of positional information of where the model thinks the functional enhancers are. Okay, so that's going to be our prediction of where is the information, where are the enhancers that the model is using to make, to predict this gene. Okay, how can we validate? So there's a nice data published by Jesse Engreitz's group, CRISPR-I FlowFISH data. So what they do is a CRISPR inactivation screen against enhancer elements for a gene of interest. So they look at, you know, for their gene, they look at accessible elements, they design CRISPR guides against those putative enhancers. They use RNA-FISH for their gene of interest to sort the cells by the expression level of the gene of interest. Okay, and then from the data, sequencing the guides in the different bins, they can infer an effect size. So inactivating a certain enhancer leads to a fold change of the gene of interest. Okay. And this paper also introduced the activity by contact score that I mentioned before, which really is just a score that uses Hi-C contacts together with DNase and HCK27 acetyl. Okay, so and that's how they do their mapping of enhancers to promoters. Okay, so what we can say, you know, ABC score does well, but we can do better when we have these GraphReg models, either using sequence or using epigenomic data and pairing that with a feature attribution like deep SHAP or saliency. Okay, so this is area under the precision recall curve for 19 genes or all combined. And you can see that the GraphReg models do better at identifying distal elements that have an effect on gene expression of the target gene, it does better than ABC, and it does better than feature attribution on dilated CNNs. Okay. Okay, just to give some intuition for why GraphReg works, okay, what we're looking at here is the MYC locus, okay? So MYC is an oncogene. It's also famous for having very distal regulatory elements, okay? So it has these enhancers that are 2 megabases away. And if you, so in orange are the predictions for GraphReg using epigenomic data or sequence data, and in blue are the dilated CNN using, the same inputs. And if you look at these tracks where you are seeing the feature attribution, what you can see is that because of the HiChIP data, feature attribution with GraphReg can recognize these really distal enhancers, okay? But the CNN just can't see them, okay? So even though the dilated CNN, is receiving this whole 6 megabase region, the feature attribution is really seeing much more promoter proximal regions, okay? They can accept a large input region, but the feature attribution is focused closer to the promoters and not this far away. Okay, I'll show you one more thing. I don't want to go over time too much. We wanted to see that the sequence model is actually learning meaningful information, okay? And we hit upon this approach, okay, where we would try to do an in silico transcription factor knockout, okay? So for example, what does GUND, this transcription factor, do to, what are its targets? Well, what you could do is find all the motifs for GUND, and in the one hot encoding, you could set all those motifs to 0. You could predict the expression of all the genes from the original sequence and from the sequence with the motifs ablated, and get a predicted log fold change, okay, from the model. And then you could ask, well, does the predicted log fold change have anything to do with the actual experimental log fold change when you actually knock down the transcription factor and do RNA-seq? And luckily, we have encode data where they did just that. They did a CRISPR-I knockdown of many transcription factors. So again, this is showing from these CRISPR-I knockdown experiments, if you look at the ones that had enough of an effect, that had at least 200 downregulated genes, you use the model with motif ablation to predict what genes would be most downregulated, and assessed, is the fold change negative, and what's the precision of this prediction? So again, the predicted genes with the most predicted to be downregulated had the most negative fold change, and the precision was higher compared to CNN models, or just a sort of class bias baseline. Okay, and just to show one picture of this, so one of the genes downregulated when you knock down GUND is this gene TCF3. So it's also predicted to be downregulated by the sequence-based GraphReg. And you can actually see that the saliency of the model kind of lines up with experimental data that we didn't use for ChIP-seq, whereas the saliency for the 1D CNN is not as associated with the actual GUND binding. Okay. How much time do I have? I can show a few more things really quickly.
Altogether, it's an hour, so we have 15 minutes. So you.
Oh, okay.
For discussion, but we can take probably some of it to present more if you like.
Okay, so I have a few things that are coming, and I'll just show some things beyond GraphReg. So just to summarize this model, this is an approach to use graph neural networks with 3D interaction data, specifically HiChIP, H3K27 acetyl HiChIP, where we can learn from epigenomic data or use prediction of epigenomic data as an auxiliary task and predict directly from DNA sequence. And we show that they outperform the kind of published dilated 1D CNN models, which are the published state of the art. So, you know, there's new work on attention-based models, though I think this approach can still see farther than the current unpublished models by using 3D interactions. And we showed that this outperforms the ABC score. And we're going to push this into more biologically meaningful contexts. So regulation of pluripotency genes or enhancer wiring and pancreatic differentiation. Okay, I show you two other things really fast. We're also, you know, there's been some really nice work on predicting the 3D contact map, the Hi-C matrix from DNA sequence. These are interesting, and they have possible uses for trying to understand how genetic variation might perturb the 3D structure. One thing is that these models don't generalize to a new cell type and don't actually seem to capture cell type specific information so well, okay? And if you're trying to predict the contact matrix for a megabase region and you're using 1 megabase, you know, nucleotide level resolution, they can be expensive to train. So we wanted to know, you know, can we train a relatively lightweight model with just epigenomic data, and can this model actually learn cell type specific information, generalize to new cell types, and can we use the predictions of the model quantitatively? Can we actually use the predicted contact map to call TADs or call interactions in a sort of statistically reasonable way? So we're working on all of this. I'll show you sort of how far we've gotten. So this is a collaboration with Jeff Bilmes and Bill Noble. And the idea is we're using epigenomic tracks. So they're actually listed here in very small print. We're using CTCF, a few histone marks, and accessibility. We're kind of using the deep sea approach of predicting stripes from an input context. But we have sort of a part, you know, a mean squared error part of the loss, but also an adversarial loss where we are trying to encourage the model to produce Hi-C contact maps that look like real Hi-C. And we're doing pre-processing with HiC-DC+ because we're hoping that we can actually do statistics on the predictions eventually. Okay, and this is a sort of an example of you know, capturing some of the major structures. What I wanted to show is just that in settings where the same genomic locus really does have a different 3D layout, okay, you know, here's in GM12878 has a strong TAD structure. It's not really there in K562, okay? From the epigenomic data, you can actually predict that, okay? And this is actually showing predictions trained on one cell line, GM12878, and prediction on held out chromosomes in, you know, the same cell type and a different cell type. And kind of getting reasonable performance. Also doing feature attribution for specific interactions, you know, and the interaction is present in one cell type, it's not present in the other cell type. And you can see that the feature attributions change, and you're not seeing sort of these the local information that might fold to give you this interaction. Okay, one more thing just as a teaser, the other thing we're really excited about is high quality multiome data, where in the same single cell you have chromatin accessibility and RNA. And that co-assay data, as you can imagine, if you're interested in gene regulation, opens up lots of possibilities of learning at a single cell, metacell, or cluster level, the relationship between the chromatin state and gene expression. So we are certainly trying to push a number of these models into the single cell realm. This is just one of many beautiful multi-ohm data sets that we're working with. This is a collaboration with Charles Sawyer's lab. So, the lab is focused on prostate cancer. And we previously showed that mutations that you see in human prostate tumors in the pioneer transcription factor FOXA1 lead to massive accessibility changes and gene expression changes in these cells. And now we can see at a single cell level that, mutations at sometimes single nucleotides, in a single transcription factor, or this, this one is a truncation mutant, massively change the cell state, and both at the accessibility and gene expression level, and also change the bias in epithelial differentiation between basal and luminal. So this is coming, but I want to put a shout out for this kind of data. I think it's really really exciting. Okay, I can wrap up here and take questions. I wanted to thank all the students, past and present, who contributed to the work. So Ali Reza Karbalegari is a postdoc. He's not on the call, he's on vacation this week, but he did all the GraphReg work. Marve Sahin graduated from the lab. She led the HiC-DC Plus project with help from Wilfred Wong, and Ray Young, together with Arnav Das, who's in Jeff Bilmus's group at University of Washington, have worked on this epiphany approach for predicting the Hi-C contact matrix. And with that, I'm happy to take questions.